Good morning or afternoon, class. I wanted to finish off the notes for Heredity so then we can move on to the next unit. But we still have a good bit of left to understand about basic genes and heredity. So the next slide, the last set, towards the end of the slideshow, is. I lost it. Dang it. Is the one on pedigree. So we have done the basic um, ideas of Mendelian genetics. We've done the irregular things like incomplete dominance and codominance. Did sex-linked traits, which is a good example of that is color blindness or male pattern baldness. And then I'm on slide 54 out of 61 slides, and it's labeled pedigree. Pedigree, yes, is a dog food, but also is a way of charting genetic traits. So think of it like a family tree, but for a family tree, we're worried about people's names, their date of birth, and their date of that they passed away. But on a pedigree, we're not worried about who's who so much and when people were born or passed away. Instead, we're worried more about did they carry a trait or not? That trait could be good or bad. It could be something mundane like could they curl their tongue? That's a trait that we could put on a pedigree and chart it through a family. Or it could be a genetic disease such as sickle cell anemia or Huntington's disease. It could be some sort of disorder. There's many different possibilities, but it doesn't have to be a bad thing. It can be a good thing. It could be a good trait. Maybe you wanted to chart how many redheads were in the family. So a pedigree uses some symbols. Squares are traditionally for males, and then circles are traditionally for females. And you'll see in that image, you'll see there is a male and a female at the very top with the line for Roman numeral 1. That's our original parents. And then they had offspring. And if you count their offspring, there are 5. You got to follow the lines because some of them married. And when they married someone, that line doesn't come directly from the parents. Instead, it is attached to the offspring from the original parents. So there's 5 offspring. The first one was a son, the second one was a son, the third one was a daughter, the fourth one was a daughter, and the fifth one was a daughter. So usually with pedigrees, there is a key. It indicates that squares are male and circles are female, but it also indicates that red is the trait that we're tracking. It doesn't tell us what that trait is, but for all intents and purposes, it doesn't really matter. We're just understanding how pedigrees work. But let's say this is sickle cell anemia, which is passed down from generation to generation. So our original father is affected. He is a red square. So he has sickle cell anemia, and he has 5 offspring, and 3 of the 5 do have the trait of sickle cell anemia. The first son has it, the very first daughter, and the last daughter, the 5th child, all are colored red, and they have sickle cell anemia. Now, that first son does pass it on to his offspring, but the first daughter, the one in the very middle, she does not appear to have married and have offspring, so she does not pass that trait on. But the last daughter, on the far end, on row that's Roman numeral 2, she is shaded red and she marries, and she has 4 children. 2 are male and 2 are female. 1 son is affected and 1 daughter is affected, but so is 1 son and 1 daughter not affected. So just to go through and make sure we understand this, let me get the whiteboard. So a pedigree, shown right here. Squares are male, circles are females, and I'm going to use the shade blue to indicate that they are affected. So our father here is affected, but our mother is not. They had 3 offspring, a son and 2 daughters. The son is affected, and he passes on to the next generation. Our daughter does not pass it on, and our last daughter does not pass it on. These Roman numerals indicate our first generation, our second generation, and our third generation. And we could keep on going it could be much longer than this. A pedigree does not have to be just 3 generations. It could be 10, 20, as far as we go back with keeping up with the data. We do use this pedigree more than just with humans. It could be used for keeping track of genes for dogs, or horses is another popular thing to do pedigrees with. Or it could even be with plants. Some people like Greg Mendel kept good journal records. He didn't make any pedigree charts that I'm aware of, but we could go back and use some of his notes to make pedigrees from his recordings. The next slide is genetic engineering. We've already covered that earlier in the course. So I had to throw it back in here because we're talking about inheritance, the traits that we get. So genetic engineering is the exception to the rule. We can engineer an organism to have certain traits. One of my favorite examples is Bt corn. Bt stands for Bacillus thuringiensis. It's a type of soil bacteria, and it has a gene in it that makes a certain protein through transcription translation that is very desirable in the world of agriculture. And a company, Monsanto pulled that gene out of the bacteria and inserted it into certain crops such as cotton and corn. So then we now have Bt cotton and Bt corn that produces a protein that they naturally wouldn't. And that protein is an enzyme that causes much damage to the digestive tract of a lot of leaf worms. So when the leaf worms start chewing on the leaves of cotton or corn, they get that enzyme in their gut and it kills them from the inside. This is introducing us, humans, introducing a gene into an organism that wouldn't get it normally from its parents. The next slide, which is number 56, gives you a little bit clarity with some terms. A genome, let me start with that one, is all the genetic material with an organism. So all the genes that an organism has. And we've been doing projects on mapping out what genes that an organism has. We've done humans, we've done dogs and cats, we've done horses, and we're trying to figure out and create a long list of what genes are available to us. Because right now, when it comes to genetic engineering, all we're really good at doing is copy and pasting. We can copy a gene from species A and paste it into species B. We're not yet able to write our own genes. But a genome doesn't tell us exactly where that gene is located on a chromosome. That's what a gene map does. It tells us where that gene is located on which chromosome. Remember, we have 23 pairs of chromosomes, so a genome would tell us what genes are in a human, but it wouldn't tell us where on the chromosome. The gene map maps that out for us. Now there's a video there that is titled, "Can we genetically improve intelligence?" I'll link that to this because it's very interesting. An interesting concept of being able to improve our species as far as our intelligence. The next slide, some results of genetic engineering may include, we have manufactured plants that make their own insecticide, like Bt corn and Bt cotton. We have made plants that don't spoil as quickly or have certain proteins that they normally don't have, or vitamins, such as our golden rice that helps boost the vitamin intake in the people that eat that rice, because normal rice doesn't have those vitamins. We have developed animals that are bigger and faster growing and resistant to disease. We have developed bacteria that may be able to help with insulin to help people that are diabetic. And theoretically, we can, in the future, fight against some of our own genetic diseases, such as Tay-Sachs disease, sickle cell anemia, and cystic fibrosis, just to name a few. Maybe we can get there. We're not there yet, but that is our hope, that we can help individuals with certain disorders with our genetic engineering. And let pause for a second. Sorry about that, I had to pause it so the slideshow would continue to load. The next slide, which is number 58, is cloning. Alright, cloning is old technology. We've been doing it for a few decades now. The Russians cloned the mouse in '86. Dolly the sheep was cloned by the Brits in the 90s. And we still are cloning today. There's universities that are cloning different species. There even was a clone of an extinct ibex done, I think, in Italy. Unfortunately, the ibex passed away minutes after it was born because of a bad lung. But cloning may be an answer to bring back some of the extinct species, especially if it went extinct within the last few hundred years. We may be able to bring them back through cloning. But cloning comes in two different forms. There's natural, and then there's engineered. That picture up there that says Wandering Jew, and it's got that purplish plant, that is a natural clone. As that vine grows, it may break, and if the stem breaks and it can reach the soil, it'll start to root. And that section, that little broken off part, will be genetically the same as the rest of the plant that it broke from. But they will then be two separate plants. That is a natural clone. There's a lot of species that can do this. For example, you may be aware of a sea star, which is not a starfish. A starfish can't do this, but a sea star is a different species. A sea star, if you were to break off one of its arms, just chop it off, and if it didn't get infected or digested by something that was hungry, then that arm can grow a brand new body, and that body that lost an arm can build a new arm. This is natural cloning that some species do. What's more interesting is probably the engineered version of cloning, where we take the genetic code from an organism and we make a copy. So we take the DNA from, let's say, the donor to make Dolly the sheep, and we insert it into an egg, fertilize it after we remove the existing DNA from it, and then coax it to divide and form a new sheep. That new sheep would have the same genetic code as Dolly. Unfortunately, with cloning, there is some shortcomings because our DNA does get wear and tear as we age. That wear and tear can pose problems. So when we donate old DNA to a organism, so then it can start as an offspring and grow and get bigger, it already has older DNA, and it's at higher risk for cancer than a regular organism, and it may not live as long. That is a problem, but there are scientists working on ways of fixing that problem. Next slide, which is number 59, is gene therapy. Gene therapy is where we take a gene and we insert it into a person or organism, so then it will then have that gene. So, for example, I mentioned earlier that we hope to do some genetic engineering where we solve some genetic diseases like cystic fibrosis, Huntington's disease, and sickle cell anemia. That would be through the process of gene therapy. Fixing a genetic problem by inserting a gene into a person that we want. Now, this is a little scary sounding because we want to use viruses to do this. A virus is very good at inserting DNA into our cells. So we could take a virus, remove its DNA, and insert the DNA we want into it, and then infect the person with that virus. And then hopefully that virus will put that DNA into the cells of the patient and give them the genes that we want. Like, for example, sickle cell anemia. If the patient has that, then infect them with a virus that would insert the correct gene for making hemoglobin, because they have a gene that is making hemoglobin improperly, causing the cells to be sickle-shaped for the red blood cells. This is still under trials. Not a great amount of progress has been done because it's still very new science. If you watch Rise of the Planet of the Apes, this is one of the premises of that movie, the very first one in the series, the new ones, not from the 70s, where they're testing on chimpanzees a virus that can fight against Alzheimer's. And then the rest is movie magic, not so much science, but we are currently working on this. The next slide, which is the last one with the notes, is selective breeding. Selective breeding is the low tech. This is old science that we've been doing now for a few thousand years, where we take organisms that have favorable traits that we like and we breed them together. Like, for example, we may have two dogs that are particularly loyal, that take care of the kids and don't bite them, and they defend the farmstead, so we breed them together to make more puppies. This is selective breeding. We don't breed the good dog with the one that is the biter. We don't take the corn that is frail and not doing well and breed it with a good one, and create some sort of mix. Instead, what the ancients have been doing for thousands of years is take some of the best, crossbreeding them with one another to make the next population for the next year or years. And this is selective breeding. This is not amazing science, and there is limitations on it. It's not foolproof. We are not genetically picking certain traits specifically. Instead, it's kind of hit and miss. But it is a method that has changed agriculture and our lives. It's created a lot of domestic animals, like our chickens and our cows and our pigs. It's created a lot of crops, like, for example, corn and wheat and rice, that are very different than their wild versions. Instead, they provide us with better sources of food, all done through many generations of selective breeding, picking the best and crossbreeding them with one another, so then we can get better and better with each new generation. All right, that closes out the last of the unit. That means we can go on to the next unit. Awesome. I'll see you later, guys.